Yeah, thank you for having me and welcome from different time zones and parts of the world. A little bit of background. As I mentioned, I'm from Darwin in Northern Australia. This little dot down the bottom here. I'm actually presenting today from Larrakia country, and I'd like to acknowledge the traditional owners of this country and recognise their ongoing connections to land and water and culture. It's a really interesting region to work in. It's a monsoon tropic climate. And it's got a reasonably diverse fish fauna, at least 1,500 species, and we're adding to that nearly every week. And it's a unique fauna just because of the biogeography in terms of being on the Sahul plate and the other side of Wallace Line. We get a lot of localized endemic and unique species, and I'll present a few of those today. You can find my publications and other things through ResearchGate and Google Scholar, and a lot of that's on gobies, but as you mentioned, a lot of it's on quite diverse fields. We're sort of trying to promote fish across a range of disciplines and areas. We do get to do some pretty exciting field expeditions in Northern Australia to really remote places in the middle of nowhere, basically. It's a big, vast landscape, lots of unexplored areas. So we're involved with the program called Bush Blitz, which is we're here out in the Victoria River, electrofishing for fishes above a waterfall. We work a lot with the marine aquarium trade to document new records. They go out and collect, and we help them sort of identify the fishes and add them to the collection, document all the different things. And that's included describing some new species of fairy wrasse, some really exciting, colourful small fish. There's the monsoon fairy wrasse and the sunset fairy wrasse and a couple of other ones that we're looking at the moment as well. Another industry partnership we have is with the fishery, the local fisheries agency, and helping them to identify catch that comes in through commercial fishes, recreational fishes, and also they have a fisheries observer program which gets out to all sorts of interesting parts of deeper water. And we run these workshops where we can identify the fish and then add again to the records of the region. We look, Northern Territory is a pretty unique landscape. It only has a handful of introduced fishes, alien fishes from other parts of the world. But just recently, so we work a lot in aquatic biosecurity, trying to prevent those infestations, but also document and control them when they do occur. So that's a big area of research in an important area like Kakadu National Park, for example. Just in the last few years, we've noticed an invasion of betta splendens, the aquarium fish, and that's starting to get out of control. You can see our paper in biological invasions. As I mentioned at the start, our work deals a lot with conservation as well. So this is a rainbowfish taxonomy project. These are an Australian plate endemic group. And we described a new species of Canzichthys from well north of its range in the Daintree River. And subsequent to that, we realised it was pretty threatened and managed to get it listed under the IUCN Red List of threatened species. So making sure that our information that through taxonomy is widely disseminated and used in management and conservation. We also work a lot with traditional owners and land managers in the remote part of the world we're in. There's a lot of Indigenous ranger programs, and we work with them in terms of aquatic biosecurity, biodiversity, two-way learning about language, and just generally understanding what's in the waterways and how to look after them. So again, another rainbowfish project up in the escarpment country around the region. Citizen science is also a bigger and ever-growing part of museum research, and it's a great way to raise awareness about your study groups and the region and the habitats and the threats they face. You can potentially be discovering new species and interacting with a key group of stakeholders who are out in the environment all the time, keeping an eye out for interesting things. So this is a citizen science project we have on worm gobies, which I'll present some information on later in the talk. So that's the research part of my role. There's also a big part of getting public programs and science communication to get that information out to the public and managers. So we do exhibitions, lots of social media, lots of mainstream media, podcasts, all that sort of stuff. If you head to the Magnet website there, you can track down a bit of that. And of course, working at a museum, we're the only major museum in tropical Northern Australia and the surrounding regions. It's a big collection, so we're always looking after the collection in the tropics as well, but also finding ways to make sure that the public understands the value of the collection and to make sure it's utilised and valued. The collection lot here in Darwin is 40,000 lots of fishes, approximately, and growing. It's a real focus on tropical freshwater and nearshore marine, but also gets out to the deep sea and well into the desert areas further south in the Northern Territory. So our collection ranges from huge marlin down to tiny little gobies. And this is just a snapshot of a few of the shelves. All right, so a bit of an introduction to me. Today I'm going to give a bit of an outline of, I'm going to quickly touch on some overview of museum science. I know we've been getting a lot of presence on that, but it's always good to get new perspectives on the collection-based research and how we might do it in different parts of the world. Then I'll give an introduction to Goby Biodiversity, and then give some specific examples of how that links to Goby taxonomy with a couple of recently published studies and a bit of a longer term ongoing project on the worm gobies. So I guess this is good perspective from my point of view about how I approach taxonomy, which might be useful for other people, but everyone's got a different interpretation. But I guess taxonomy to me is about classifying, trying to get names on things and making sure we understand what part of the tree they live in. A big part of it is trying to look at discovery and revising taxonomy, and just discovering new species, revising taxonomy, and getting actual formal names on those species so that they can be known. If they don't have a name, typically it's very hard to manage them. But there's a lot of obviously long-term problems to chase down in the nomenclature and naming of things as well. It's also really important to not just name things, but give useful tools for people in different stakeholder groups. So all the way through from managers down to aquarium keepers, researchers, to be able to tell the species they have apart. And that's a really important part of modern taxonomy, is to make it very applied so people can see the value of taxonomy. Taxonomy has been in a decline overall through museum science, and we need to really reinvent it and keep up with the technology and the tools available. Why do we even care about species at that level? I guess they are pretty interesting. It's a really important part of biodiversity and unique variation and evolution and adaptation. But it can also help us to make sense of broader patterns. So whether that's biogeography, our local region, the food webs that occur in there, we need to know how they're structured. And also different species do different things. So it's really important to tag them so that we can then follow that through the ecosystem. And accurate biological data helps to really inform fisheries management. So if we don't have good taxonomy, we can't get good identifications, we can't get good biological data, and then we can't manage fisheries, and we might be losing species before we know they exist or before we realise they're gone. And to link this back to museum collections, I like to use this acronym with JAR. So it's a journey through time. It's there for the long haul. They're permanent records that can be used by a wealth of people. They provide accurate data because they're, and I'll talk a bit more about this in a minute, they're defensible and verifiable, and they've already been collected. We're not guessing where they occur. And they're also a really strong resource for the future. We can always review the collections as technology changes, and it's particularly in modern times when resources are very limited. Previous collections in different times are a huge bank of time, energy, and expense. A lot of the collections, like in our collection, we could never recollect. So it's pretty priceless. And I guess one of them, again, the modern challenges to collections-based research and taxonomy is the notion of cryptic species. It's a modern, all-pervasive problem that occurs in all regions, all habitats, all animal types, and particularly all fish groups. It really challenges what we know about the fauna, especially in regions where the fauna is not very known or there's high biodiversity already. And that can just confound out the ecological data that we're collecting. So we might not be able to pick patterns out of the data because there's more than one species involved. There might be two different peaks that makes not much sense because there's more than one species involved. Or we actually might miss some really rare or species that are doing different things to the norm, and they're the ones that can fall through the cracks and disappear. And it's a special problem for groups like gobies that are small, less colourful, and I often get it, and you probably, as you mentioned at the start of the lecture, people don't really pay much attention to gobies. They're not big, you can eat a few of them, but I always get, gobies all look the same. And it's like, no, they don't. Once you start looking hard and really get your eye in, it's just that you can get overwhelmed with the diversity of gobies. So don't think like that, drill in and get to know gobies because they're pretty cool. And a big part of collections-based research is having the vouchers. Getting the vouchers into museums is the real challenge and the most important part of taxonomy today, so that you have something to go back to. And I'll talk a little bit about the processes we use to link these vouchers to all sorts of data. In terms of taxonomy, the modern approach now is to use combined lines of evidence, not just to rely on one particular type of data to define species boundaries and characters. So the work we're doing now really looks at morphology, so the traditional taxonomy, doing counts and measurements of specimens, but also to couple that with different perspectives from genetics. So barcoding is one type of genetics, but also particularly with freshwater fishes and around Australia, we're finding that there's a lot of hybridization and heterogression. So using multi-locus nuclear markers is a really important part of how to understand the taxonomic framework. But the combined approach of nuclear and mitochondrial gives you a really good answer a lot of the time. And then applying another filter or level of understanding to the taxonomy and species is looking at their ecology. So how often they look different, whether that doesn't show up in morphology or they behave different, they might have different biology, and we can link all that together in a species description to help tell them apart. And I like to think that collections are like a library. They are a real important reference collection where we can go and look at animals within a dedicated facility. Specimens and tissues are available for research to other researchers, and we take special care of things called types, which hold the name-bearing types of the animals. And it's a real base for collaborations. People come to visit us, we send out loans, and that way discoveries continue over time as there's so many different fish groups in the world that different experts come through and have a look at different things. And it's all part of the bigger picture, understanding having these collections helps to provide feedback and value adding to different stakeholders that contribute the specimens. So it's really important to feed back the information in the collection to public databases or directly to stakeholders. And this is an important part, I guess, for people especially that do environmental impact statements or assessments, that fish collections, rather than just having data or grey literature, are a really important part of quality assurance and quality control. A voucher is a physical permanent point record. It can be checked in cases of confusion or controversy, and it can also be re-examined as information and technology changes. So as taxonomic revisions occur or DNA studies are happening. So the voucher is, again, I'll harp on vouchers, but they're really important. And preparing the vouchers, I like to call the different approaches, but if you have the time and resources, I like to use the ultimate voucher method, where you get the most information out of and most value out of a single fish. And this is really important from ethics these days as well, to make sure we're reducing and refining our approach to the numbers of fish used. So the voucher itself is pinned, photographed with its fresh colours, and has a tissue tag and a tissue taken that travels with it for the rest of its journey. We can then identify the specimen using library keys and our network of experts, which it's great to see so many people from different parts of the world today. And one thing that's really important with the specimen is its data. Make sure we get the data, register into the permanent collection, and then serve that data so that people can see it and understand. And this is just an example of a field sheet that we were using for some deep sea fish the other day, linking it all back to this individual unique number. Pinning the specimens makes a huge difference to the taxonomy and presentation of the specimens for when you see them in the collection, and also to the care and preservation of the fish. So you can see the difference between two ambassadors here, one without the pins pinned out. We then would paint the fish with formalin to make the fins stay there, remove the pins, and then we tie the individual tag or attach it to the fish so that it travels with it. Getting those life images really helps to sell the value of the fish. So we've got different techniques. This is one for small fish in a Petri dish. But we've also, thanks to Deepak, who's a research associate in Darwin with me now, he's videotaped a recent processing workshop that we did with fisheries, and we'll make that video available to participants through the workshop. I'll give you the link at the end of the presentation. And that talks through the whole process from data, tissue sampling, specimen preparation, photography, and fixing as well. So that's a really good resource. Thanks, Deepak. All right, so let's move into gobies. They're part of a really large order, and I'll refer readers to a whole heap of studies that are underway and ever going. The relationships where gobies sit in the family and higher level and between families is still a lot in flux. So I'm just going to specialise today on the, as we currently know, family Gobiidae, with five subfamilies and about nearly 2,000 species, and it's ever growing. It is the world's most diverse fish family by that definition, with an almost global distribution across nearly every sort of habitat you can think of, from coral reef, sand, deeper water, shallow estuaries, right through into the desert. You can see, yep, they win the species challenge by family by a little way from some of the more other well-known groups. So yeah, we do need to know more about gobies because they're a really prominent part of most ecosystems and habitats around the world. So from that point of view, they are a pretty important part of local biodiversity, and there's a lot of narrow range endemism in the groups, whether that's a coral reef goby or a desert fish, through to estuarine species. So that gives it a regional meaning. Some of the larger growing species like the Pseudogobius and Amblyopinae are certainly used as food fish in some areas, whether that's in larger fisheries or more traditional market-based fisheries. They're a big part of the aquarium trade, whether that's reef fish or freshwater and brackish trade. Some of the smaller species are really specialised and suited to nano aquariums. They're also, because of their abundance and diversity, are good biomonitors in particular areas, particularly estuaries, and model organisms for understanding environmental change. And they're also an important part of culture and language, particularly in remote areas. So that general abundance makes some important links in food chains and ecosystems, especially on coral reefs. There's been a lot of studies showing that some things like Ichthyotaxonomy are the basis of food chains. So yeah, the more we understand about gobies, the more we can understand about ecosystems. And to me, the huge diversity in form, some species like shrimp gobies live with a mate down a burrow, or a blind goby that has no eyes, or another one that's jumping around on top of the land as a mud skipper. It makes it really fascinating to see the diversity, how it unplays in the landscape and how it might have evolved. So let's break the gobies down, Gobiidae down into the subfamilies of all these weird things. And this is a real simplification. Each subfamily in its own right is very diverse, but we have start with the Amblyopinae, the blind worm gobies that mostly live down in the mud. The mud skippers that have kind of gone the opposite and gone above the mud and live most of their life out of water. The true gobies, which is a really diverse and large subfamily, things like Glossogobius through to shrimp gobies. Gobiidae, which is a really diverse group that I've been working a lot on with Dr. Helen Larson of late, looking at things like Pseudogobius. A lot of things that live in the mangroves and freshwaters are in this group. A lot of localized endemism in there. And obviously the stream gobies is another important group as well, particularly in high rainfall areas, offshore islands, etc. I just put some little stars there. These are 3 groups that I'll be giving the examples taxonomic studies of today. So the Amblyopines, the Glossogobius, and the Pseudogobius. And just to break down the subfamilies, you can see that, yeah, they vary a lot, but also I think the last column there just shows how much species discovery is still occurring within the Gobiidae. It's 300 species in the last 10 years. I'm sure that rate is not going to flatten off anytime soon. All right, so let's focus on some of the things we can do to, if we want to study gobies, either just to identify them or to then go into more detail to start taxonomic revisions. By and large, most gobies are small. So there's going to be some general rules for dealing with small specimens and small species. We need to be extra careful in how we collect them, firstly, so the types of nets, the way we net them, and how we handle the specimens after we've collected them as well, before preservation. So in that sense, once we've collected them and looked after them, got them back safely or in the field to a base camp or back to the lab, there's going to be a balance between trying to take a tissue sample off an animal. So you might be trying to fill up the right-hand side of a fish to get a little muscle sample off. How much that will actually damage the whole fish by handling it versus the type of fixation you can then apply. So ideally, we use formalin fixed material for studying the taxonomy, but sometimes we have to maybe fix the whole animal in ethanol so that we don't end up damaging to get the tissue. So there's all sorts of combinations of that, but it's just to keep in mind that preservation will be affected by the size of the fish. And if we do fix them in ethanol, it can make it pretty hard for trying to get fin ray counts and other things because they become so brittle. So often a combination of both works well. It helps to know your group. So all the different genera of gobies typically have some unique features which help to a diagnostic for different species. So particularly around the head, we've got a lot of gobies that have barbels, papillae, other features on their head that are really important. So we often, when we're preparing the samples, we're often very careful around the head and not to damage it, but also things like Glossogobius that have papillae that will shrink down. In general preservation, you need to paint them fresh with formalin before they can shrink away. So just little things like that. So that's an adaptive process as you get to know your group more. And what we're finding, I guess, especially with cryptic species, is that color becomes really important. I guess they need to tell each other apart. So we're trying to cue in on the different visual cues they might be using, and that can really help us with the taxonomy. And so getting life images makes a big difference. So whether that's an in situ scuba shot, a field aquarium or a longer term aquarium, or some freshly preserved specimens in a tray, as I showed earlier. And it is worth going to the effort to spend a bit of time pinning out the fins from this point of view as well. Like with a goby we recently described with Doug Hosey, just pinning out the dorsal fin was a big help because of the big black mark that distinguished it from a related species straight away. If the fish had its folded fins up, it would have been hard to use that as a character. I just provide some examples here of different methods of photography of these fresh life images. So some of these, this little Ichthyotaxonomy was taken in that setup I showed with the Petri dish. This is more a field aquarium shot of a goby. Here's that Tommy Micheal looking at the color pattern, particularly in the dorsal surface there, and other fish prepared in the same way in a flat tray on a neutral color background to make sure you see the most different colors of the fish. This is a bit more of a long-term aquarium subject to get it in its, when they first come in, they're very dull and gray. It takes them a while to get their colors back to then see how they present. A couple more examples, again, just to show the diversity of gobies, but also different methods of documenting their life images. So we've got a nice Gobies here, trying to get the cheek patterns and the caudal markings to show up. They often just go black in preservation. Yeah, the colors on this guy are pretty amazing. Underwater photo of one of our local gobies, and getting the whiskers in this guy really makes a dramatic photo. And obviously microscopes are a big part of work on gobies. They're tiny, but they do help find a whole heap of different features. So places to look, obviously, are under the chin for little frenum, barbels. This is the typical disc-shaped pattern of the pelvic fin in the typical form in gobies. We're then looking for scales on the cheeks and the operculum, different rows of papillae, different transverse or longitudinal rows. Again, really important as part of keys, raised ridges, other features of the head. So just some example shots, and I'll go into a little bit more detail with the specific studies in a minute. So I guess that's a quick overview of Goby taxonomy, but the real way is just learn by trial and error in a lot of ways. And I'll show you some trial and error studies now. The first one we look at is Pseudogobius. These are Gobiidae, a small fish up to about 5 centimeters at the most, grey colour. When you catch them in the wild, they don't really look like much. They're sort of grey and indistinct. It's not till you get them into a fish tank or into properly prepared that you can really start to see their markings. They're distributed very widely across the Indo-West Pacific, and they're really an abundant group. Pretty much anywhere you can go in the region, you go into a little shore pool or into the mangroves, you'll find these guys. And in some areas, they're really abundant in seagrass meadows and things like that. There's, like most goby groups, there's been really a historical confusion over the names of different species and how to identify them. And that's not common, not just only to Pseudogobius, but it's common to most small goby groups. And it's taken a massive amount of work by Dr. Helen Larson to find all the old names and see how they relate to look at valid species. Our taxonomic approach was an adaptive feedback loop. So starting with morphology and then incorporating some genetics and then going back to re-examine the morphology as an adaptive feedback loop to see how it all links together. So we've just published a major molecular revision on the group focusing on species boundaries. And that's complemented with a taxonomic revision that's just a week or so away from being published. So the focus for the molecular part of the work was on initially on intensive sampling around the Australian coastline. That's several thousand kilometres of coastline just for scale, so about 5,000 kilometres. We got to most of it. Some areas are natural gaps. This is the Great Australian Bight. There's no real estuaries in that area. And likewise, this seems to be a fairly natural gap in the group because there's not a lot of fresh water through that part of Australia. We then reference that to available samples from other parts of the world. You see there's not much Indian material in here in GenBank or other places, so that can be a future target for another regional study. And so this is a bit of a summary of the genetic patterns, reference back to geography. You can see the first real pattern is this is the Tropic of Capricorn through Australia, the tropical north, temperate and cool temperate to the south. There's a really clear divide between the tropical fauna and the temperate fauna. There's a little bit of overlap on the side here, but yeah, so you can see that's the first split we noticed. And then more broadly, we're seeing a big split in the species that occur in Australia versus east of the Lydecker line. Traditionally, Pseudogobius in Australia, there was known to be 2 species with a few other cryptic species touted. But at the end of our molecular study, we identified 9 species occurring in Australia. So that includes a lot of cryptic species and localized endemics as well. So the significance of the study is high, and it's not unrealistic that lots of other groups will have the same patterns. This is a tree summary of the nuclear data. This is an example here of the adaptive feedback loop. So as we collected the animals, we assigned a working operational taxonomic unit or just a morphotype to them. And then we were checking that back against the genetics. We had a patterned morphotype that ended up actually being 2 different species that aligned to 2 different geographic areas. So that's a real example of how that adaptive feedback between genotype and morphotype can work. Once we started looking hard at the data, we realized that it's quite easy to separate the species based on good photos using the patterns in the caudal peduncle. Some do not have any patterns. Often there's a dual pattern. Other markings on the head and the body help distinguish species. So we've got a stripe on this one through the eye. This one's got a yellow half moon shaped tail and its crescent shape in its tail. And then the fin markings. So these ones have lines of stripes versus scattered spots or no spots. So, you know, they're very basic visual characters that we use to set up our morphological framework. And overall, we found that Northern Australia was like a hotspot of cryptic diversity. Southern Australia was much more complex, and this is the pattern that we need to be very mindful of with freshwater fishes and obviously now estuarine fishes. Freshwater fishes are often subject to isolation events in evolutionary time, and it's just a matter of how long those populations are isolated to whether they become reproductively isolated or whether secondary contact might then remix them or it might lead to reinforcement. So this is an example where both modes were occurring in Southern Australia. We ended up from our initial molecular analysis of a couple of hundred fish for allozyme loci, 50 allozyme loci. We ended up with 3 primary clusters. Within those clusters, there was 2 groups of differing types of admixture. We then went into a population study to explore this type of admixture because it looked much more clinal than these were more neat F1 hybrids between 2 much more widely spread species. So if you then apply that to a geographic map, you can see we had an eastern and a western form. Within the eastern form, there was 2 subgroups, but with really strong integration across them. And we concluded that they were probably historically isolated, but have not had the chance to become reproductively isolated. So we concluded that there's likely 2 species, maybe 3 species. And then we were going to seek extra information from mitochondrial DNA and morphology to get that combined lines of evidence approach. Okay, so this is our, just to swap back to this quickly, we did find that there was only 2 mitochondrial lineages, but the boundary between the genetics, the nuclear genetics and the mitochondrial data was not very well aligned. The mitochondrial lineage of East went all the way over here. So there's been some sort of historic process of selective sweep or movement that has masked what a true species boundary was. And that is really important from a barcoding perspective because the barcoding won't give you the direct answer the nuclear information will. So we just look a bit more widely at the Indo-Pacific region. As I mentioned earlier, there's a major break across Lydecker's line, pretty much the Australian plate and everything to the west of that. And whether that's in green here, I've highlighted all the lineages that come from the Australian plate. This is now our mitochondrial data to reference more broadly. And even within, so a lot of these are localized cryptic lineages, except, and then other ones, there's more widespread taxa that have Australian lineages as well. So you can see that it applies to endemism, but also to more widely spread groups. And then we need to then expand the study to much more regional, other regional populations to see how those other patterns might play out. There is some confusion over what names to apply to what species, particularly as there's 3 type localities for very similar animals within about 50 kilometers in central Java. So that was a question for the morphology. From the genetics, we didn't find any evidence of those species moving into Australia. I'll just introduce, this is Helen here. everyone would probably, that knows Gobies will know Helen. She's been working in the group for a long time. She's been looking at Pseudogobius collectively over at least 30 years. As she visited a lot of the museums around the world, she would look at the types as well as other material and start to tag Pseudogobius for inclusion in broader morphological work. So I think at the museum at the moment, we've got nearly 1,000 jars of Pseudogobius from across the world and across a lot of institutions to try and solve this issue. I came along maybe 10 years ago and helped Helen to refine the taxonomic framework by just from in the field, seeing different color patterns and different observations, and then using that to inform as an adaptive feedback to inform the morphology to get a much more robust and holistic taxonomic framework. So Helen pretty much went by looking at all the old names and all the old material, deducted that there's 21 nominal species. We concluded that there are 15 of these are valid, including 7 species that need a new name. The morphology had strong support with the nuclear data, suggesting that there's only 2 species in Southern Australia, and Eastern and West, without any real characters to separate those 2 subgroups in Eastern Australia. And of the 3 Java names, Poecilosoma is the oldest, and they all appear to apply to the same animal. So as part of the morphological revision, a whole heap of 80 odd pages of taxonomic joy, we've described the species in detail, made lots of comparisons to be able to tell them apart, mapped all the distribution based on the records and the specimens that we've examined. There's some examples here of different taxa, and then include a whole heap of photos of different photos, so life images, preserved specimens and holotypes, so that different people can interpret it and use it in their collective ways. Yep, that way. So this is just a bit of a picture summary, a summary of the 9 species that occur in Australia that we know now. And as you say, if you hark back to the earlier question, all gobies look the same. Well, they might to you, but to me, I can tell them all apart. And like most groups, the more you look at them, the more you can start to see how different they are. But this is a great example of a cryptic species complex and how you need to have multiple lines of evidence and how important voucher specimens are to unravel these problems. So we've managed to have a major expansion of knowledge on the group, better understand what patterns are driving or processes are driving the patterns that we're seeing within this genus and also in other estuarine biodiversity. Really flagged the issue of cryptic biodiversity as an important one locally and globally. And we can sort of see that Australia, we ended up with 7 new taxa, or it was actually 8. One of the ones that was supposed to be here wasn't here. So what's going to happen when we do these really intensive studies in other parts of the world as well, using the same approach? So we've got a framework there, and now we can go off and other people can look at their local fauna. And I think it really demonstrates that we don't really know much about all our Goby groups. So the other Gobiidae groups like Micheal, Hemigobius, Pandaka, they still need a lot of work. Gobiopteris, I'll throw that one in for Kevin. All right, so that's Pseudogobius, a quick rundown of that. Let's have a look at Glossogobius, which I know is a much more well-known group across the world, particularly India and Bangladesh, where they are really sold and utilised as food fishes. But they do occur very widely from, across the offshore islands all the way through the Indo-Pacific. And often they're quite abundant where they do occur, whether that's a little bit further inland. They're not often common in estuaries. They're more common in the lower reaches of freshwater at low altitude. Some places like in India and New Guinea, they do stretch right up into higher elevations at several hundred meters. And there's a general similar body form. That means that most of these different species are, to the naked eye, are very easily confused. In Australia, we have 3 very similar looking large growing, up to about 25 centimetre, 30 centimetre growing Glossogobius, the Aureus, Gyurus and Monroei group. And traditionally researchers have had real trouble telling them apart. So we were getting a lot of feedback from collaborators and other researchers about, hey, can we solve this problem and look at fixing them up? So we started a project in collaboration with Charles Darwin University, the Australian Museum and South Australian Museum to really see if we could nut out what was going on. Again, we used this adaptive feedback loop between our field IDs and other characters, the genetics and the morphology. The genetics has just been published as part, including a field key, like for researchers. And Doug Hosey and I are currently doing a small taxonomic revision to put names on the Australian ones, but also to provide the broader international context. This is a typical field site for Gobies. This is the Daly River, a large perennial river, lowland river, this section anyway. So our point was to go and catch a whole heap of Gobies as part of, this is an long-term environmental monitoring program where they catch a lot of Gobies. So there was specific data to then feed back to the researchers about what they were catching. And this particular site, we ended up catching 4 sympatric species where they were identifying 1, maybe 2, and all the names they put on were wrong. So just shows the real challenge and the value of taxonomic work. So we did this local study, but then there's also a whole heap of exciting barcode information. This is a truly global study just based on the amount of other labs that have got involved with Gobies barcoding. So we were able to use that to put our study into a bigger reference. Again, we started with the multi-locus nuclear markers as our preferred way of defining species boundaries, independent of any hybrid, or to flag any hybridization introgression. But luckily we got 4 clean groups this time with really large fixed differences. So different combinations between all of the different groups. So these are 4 very different animals. You might say 4 to 5 fixed differences is normally used as a good yardstick, depending how you apply it. These things are up 16 plus. So good species, no issues there. What we did find was there's quite a lot of regional substructure between the different groups, within the different taxa. And we also identified that there was 2 taxa within what has been the morphological description of Gyurus, which relates to the number of papillae rows on the cheek. So we then fed our initial genetics back into the field sampling, collected more samples, started to look at the visual characters, fed that back into the genetics, and it's a big evolving cycle to eventually work out. We had some 4 phenotypes, if you like, that we could readily identify and apply characters so that we could develop a key. Really importantly, from a geographic and management point of view, these things all lived in different areas. If we put the Daly River catchment on there, you can see it seems to be a natural junction point between different species. We did get all 4 species sympatrically there at the same location, but in general, Aureus only lives east of that catchment. Same with Gyurus A. Gyurus B is an inland, much more upland species that lives through the Kimberley and the Pilbara. Monroe is supposed to be an estuarine species, but in contrast to its known ecology, we found it many hundreds of kilometres inland in lowland systems as well. So not just we found their distribution, but the initial genetics taught us a lot about the ecology. Gyurus A was much more of a coastal species living in perennial systems. And this is how we turned it into a visual character. Simple characters looking at, again, with well-preserved specimens, which you can do in the field by lifting up the fin rays and fin spines, looking for spots that are on the start of the first dorsal fin. So the 2 Gyurus have that spot. Aureus has a spot at the back of the fin, mostly in males, sometimes in females. And Monroe tends to have only a diffuse spot through its fins. The body stripes are most prominent in this form. And then under the chin, we found there's quite different coloration patterns too. Monroe actually has these little lobes, which you can sort of tease apart quite easy with your thumbnail to see if they're there. This Gyurus has black marks there, and the other 2 species are plain under the chin. And that was consistent across a couple of hundred samples. So we're confident that they're good field characters so that we don't have to then sample all Gobies into a jar. We can now selectively sample and also get confident field identification. So that's a win-win for taxonomy, but also for the ecology. Okay, I thought at this point, we thought we were doing pretty well. We'd sorted out Australia, got a couple of different species. But then when you dived into BOLD and GenBank, and it is a really amazing group. There's such great coverage of this across the Indo-West Pacific through to South Africa. We figured out there was actually a couple of additional, so our 2 species in Australia don't just occur in Australia, they go south. And I'll just emphasize here before I say species that we haven't been able to test the nuclear genetic markers across this spatial scale. So I'll just call them we found the same lineages globally, but that we need to test this further with nuclear markers. And that also links back to the morphology. We found different patterns in the morphology than to what the mitochondrial says. So watch this space, but yeah, we did find a lineage that occurred from the Kimberley all the way through to South Africa and India and Bangladesh. This Eastern Australian species doesn't just live there, it occurs all the way back the other way. So, and then they, again, all 4 species co-occur. Munro was an Australian endemic as far as we can tell. And then there's a couple of additional species currently lumped in the Gyarus complex through India, and another one down in South Africa. So I'm sure as more regional studies are conducted, and our study hopefully provides impetus for future barcoding, and other studies will be able to better map these out. So overall, there's been a real improvement in understanding of the group, particularly in Australia. Now that we have a field key, we can keep working on it. And we've provided the manuscript we've got now is providing some placeholder names for the Australian ones. There's some, so we can have a name for the 2 different ones in Australia until the broader taxonomy can be solved. The broader taxonomy needs a similar study to what we did, where we've got the combined lines of evidence using nuclear, mitochondrial, and phenotypic and morphological data. There are heaps of old names of Glossogobius stretching back to the 1800s, and most of them are really faded. They have no color markings left, they're just brown. And so that makes it really difficult to look at these characters to see which morphotype matches to genotype. So some ancient DNA studies may really help to match the type names back to candidate species and help solve the broader taxonomy. That's just a close-up of our Glossogobius pinned out, ready for examination. So a really interesting group and still much more work to be done. But I'm glad we've managed to solve it in Australia for now. Let's move on to the blind gobies. These are really weird and wonderful critters. This is a photo that Barry Russell, and also emeritus curator at a museum here in Darwin, took in Hong Kong. So again, another species where in some parts of the world they are used as food fish. Just depends how hard they are to catch. Most of them live down burrows in soft sediments and only move around at certain times of the year, potentially. So these gobies, unlike Glossogobius and Pseudogobius, which are pretty easy to catch and really are well represented in collections, worm gobies, and particularly some genera, are rarely seen and very poorly represented in collections. Historically, there's been just massive confusion about their identification and their distribution and the names to be applied to them. But thanks to about 15 papers in 20 years from Ed Murdy and colleagues, we've made some real massive strides in how to tell them apart. There's still a bit more to go, but yeah, it's an amazing volume of work if you just go through and search up those references on Google Scholar or Ed's ResearchGate page. My involvement with blind gobies has just been a long-term project to accumulate Australian samples for morphology and genetics and live images. A lot of the specimens, because they're so rare, most of them are only known from specimens. So again, a live image helps to tell them apart and promote them to the community. We've also got that citizen science project where anglers might only catch once in their lifetime, but there's enough anglers out there that we're getting a steady trickle, maybe one every couple of months, either a photo or a specimen of these things. So that's the scale of what we're up to. And it does appear that Australia is a real hotspot for this group. There's lots of different species and genera in our waters. So today I'm just going to present this group as a bit of an example group for Goby taxonomy in general, and some of the characters you can use and approaches you can use to tell genera and species apart. So X-rays are really important for this group. And I know it's hard to get good X-rays in some parts of the world, but the effort, if you can get to them, they really help you to tell, especially at the genus level and subfamily level, where they belong. Zoom in a bit here. This is a fish named after Dr. Larson, Tripalconychthes from, it's a deeper water species that occurs around offshore NT and all the way through to the Philippines. And this group of gobies, which is called the Tripalcan group, has a specific formula in the dorsal arrangement of pterygophores here and dorsal rays elements. So this is what we call a 3-1-2-2-1 formula, and that puts it into that group wholly. The next thing we might look at is the different types of vertebrae. So here's the caudal vertebrae and the pre-caudal vertebrae after that. One of the genera in this group can be defined by the number of caudal vertebrae, sorry. And then let's look at the pterygophore patterns, which are the bits that hold the fin rays up, or the fin spines and rays up. And different patterns in different parts of the fish. So in between these neural spines and hemal spines, in front of the hemal spine, you can see the numbers of fish and their pattern, numbers of pterygophores and their patterns are diagnostic. So for Amblyopines, as a group, having most of them anyway, having the first two pterygophores of the second dorsal fin in a single sharing in the same interneural space diagnoses them as an Amblyopine. Other patterns for the anal pterygophores, it can help to tell you what species or genus it belongs to. And then when you combine that with vertebral counts and counts of fin elements, you get a diagnostic set of markers to tell each other apart. But external features are pretty handy as well. You don't always get below group, but it does tell you where you start. So these is one of my favorite features of worm gobies. Some of them, just in the Tripalcan group, have this little pouch, and we don't really know what it's for. It's never really been solved what it might be used for. Maybe it's gas exchange or it's a leftover from something else. Likewise, this one here doesn't even have an eye. It has a socket where the eye used to be. But there are often some that have really reduced eyes that are good at photoreception, I guess. Scale patterns are important to a lot of the species. So this one has a naked belly versus another one that might have scales, large scales fully across the belly. You can't see the fins here, but the pattern of the pelvic fins also helps to place it into genus or group. So whether it's rounded, partially separated or fully separated. This is another type of, another species in the Tripalcan group, and you can see it's got this really cool mohawk up the top, a serrated crest, which some other species have one, but not to anywhere near as prominent as this species. And you can see its little eye under the skin hiding in there. So it can't be very well developed. Best it could do would just be able to tell night and day. Other species in this group have huge fangs. There's another way to tell them apart. So this is Amblyo Tripalcan from Eastern Australia, the Great Barrier Reef area. Just some, I said, these, they don't look much. They look pink and worm-like, but they have a whole heap of interesting features. Another one in this genus, in this group, Tripalcan group, is Gobiidae. And again, it's one that doesn't have a pouch. And then once you get to that level, you can look at how far the scales come up, whether it's fully scaled or half scaled to place it into different species. Let's move back to X-rays again and to a different group within the Amblyopines. This is the Taenioides group, so a group of long, skinny worm gobies. They have a different formula, so you have to add another empty space at the end. So their formula is 1, 2, 2, 1, 0. And this particular genus is very distinctive in this Y-shaped second anal pterygophore to help diagnose the genus. But the outside is pretty handy too. If you look at this as a fresh photo from pretty much caught out the front of my work in Darwin Harbour. On the mudflats on a super low tide, we have 8 metre macro tides in Darwin, so it can be 0 to 8 metres in a day. And you can see this fellow has got some interesting raised ridges on the head with little papillae. And that is diagnosed as the genus Taeniortes. Then looking at the patterns of these raised ridges helps to get you to species, or at least get you to groups of cryptic species. This genus needs a lot of work to solve the global number of species and identity. Barbels are also on the chin. You can't really see them that well here, but there's different patterns of barbels under the chin that help to tell different species as well. Let's have a look at another species in the group, Triporchinopsis. Let's look at X-rays. This is cleared and stained, so a different way of looking at the autonomy. Most people are now using micro CT scans, which is much more fancy than what we have in Darwin, but still pretty handy here. So you can see the last pterygophore is forked in this species. Most others will only have the single element to it. All right, within the Amblyopines, there's also a group of atypical species that don't really fit the morphological criteria of Amblyopines, but still look like those white blind animals that fit in the Amblyopinae. This one here is a very rare animal known from only a handful of specimens, Pseudogobius. It's got these really long pectoral rays, which help to tell, you can tell that pretty instantly what it is. But once you X-ray it, it's got a different arrangement of pterygophores. Instead of being both in the same neural space, they're either side of the spine. So it's a pretty fine detail, but it's an informative character and they don't seem to fit in. It'd be really interesting one day to see how genetically this matches up to other Amblyopines and related subfamilies. This is a weird fella. This one came from, I think, 80 meters offshore the Northern Territory. It's only recently been described as a genus has only recently been described. And it's got a weird little character here with these little funny eyes. As I mentioned, most Amblyopines have small eyes. This one's got big, giant moon-like eyes. Just a bit of a quick look at the diversity. So overall, Amblyopines are a great group to learn your craft, especially if you've got access to X-rays. Because they're so diverse, you get some good characters at the high level, and then working that back down to the species complexes. I'll just finish up now with some conclusions from the work. Really, my main message for today is that good taxonomy promotes good science, and that permanent voucher specimens are critical to this good science and good taxonomy. Nothing else will work unless you start from there. So collections-based science is a foundation for sound management and conservation, and we really need to start working to improve the reputation and the activity of taxonomy around the world. Gobies are a really large group, and they're still going to grow a lot. So there's an ongoing and probably perennial need to study the taxonomy of the group. The small size of some groups makes them a real challenge, but I've hopefully highlighted some techniques, and I'm happy to chat to you after about other techniques to really delve into the species level. One thing that's really noticeable from our studies was the wide distribution of these groups makes them ideal for international collaborations or international data sharing through projects like the Barcode of Life. That made a huge difference through the Gobies project to be able to compare samples from across the world. So in general, keep an eye out for gobies when you are doing fieldwork. Hopefully you think they're a little bit more interesting now, and you might keep some for your collection and start to share and collaborate the information to get a better handle on the species. I've left my contact details here. We'll leave this slide up. And this is the link. If you type that link in, you can watch, there's about an 8-minute video on that Deepak put together on our specimen processing. So with that, I'd like to thanks for your time.